My name is Nick Miller, and I work for the global NGO One Health Alliance, One Health organization. The focus of this alliance is to mitigate the spread of disease to humans and wildlife through vectors such as insects. The One Health Alliance is a global organization. I myself am one of the specialists here in Pennsylvania. Today I'll be presenting on the subject of tsetse flies, more specifically a particular species of tsetse fly, Glossina fusepes. This presentation will cover both a detailed description of the species of fly and the dangers it can pose to us, and One Health's approach to the control of the vector. So what is One Health? The One Health Alliance, or OHA, is a global non-governmental organization tasked with mitigating the spread of diseases to humans and wildlife through vectors. As a research scientist, I work to achieve the OHA's mission by studying the causes and patterns of human disease. In doing so, I examine statistics to identify health threats and recommend strategies to the OHA that could reduce these threats. Like I said, I'm a researcher working with OHA on the subject of vector biology. Vector biology is the science of understanding the biology of vectors, how they transmit diseases, and how they interact with human and wildlife hosts. You might be asking yourself, what's a vector? Well, vectors are living organisms that carry and transmit infectious pathogens into other living organisms. Common vectors include mosquitoes, ticks, fleas, mice, or in my case, flies. Research of this science is important because it can ultimately lead to the development of ways to prevent the transmission of vector-borne diseases. And vector-borne diseases are dangerous, accounting for more than 17% of all infectious diseases and causing more than 700,000 deaths annually. Infectious diseases like typhus, malaria, sleeping sickness, and Lyme disease can all be traced back to specific vectors. OHA-conducted research can help mitigate the effects of these diseases by identifying the vectors prevalent in a particular region and notifying the people of that region to preventative measures. Researching vectors has provided us with the ability to alert people to the dangers of vectors in that region and advise them to participate in the preventative measures like wearing thicker clothing and using insect repellent. Understanding the dangers that vectors pose to us is half the battle. The other half is properly identifying those responsible. Our presentation today covers an infamous species to the people of sub-Saharan Africa. Glossina fusepes is a species in the genus Glossina, which are commonly known as tsetse flies. These flies inhabit the high humidity areas of sub-Saharan Africa, with the subspecies living within the very southern part of the Arabian Peninsula. All species of tsetse flies are believed to be capable of transmitting pathogens, but Glossina fusepes in particular is one of the major vectors of one particular pathogen that is especially dangerous to people in the region, but more on that later. These African flies can be observed in three distinct forms: larvae, pupae, and adults. The adults, like the one pictured here, are relatively large flies, reaching lengths of 0.5 centimeter to 1.5 centimeters, and having a recognizable shape which makes them easy to distinguish from other flies. This recognizable shape exists within their large heads, unusual antennae, and distinctly separated eyes. Not to mention their thorax, the middle segment between the head and the abdomen, is quite large, while their abdomen itself is wide rather than elongated and shorter than the wings. They also have a distinct proboscis, a long, thin structure attached to the bottom of the head and pointing forward, and folded wings that when at rest fold one on top of each other. The life cycle of a tsetse fly is quite an unusual one. Female flies fertilize only one egg at a time and retain their eggs within the uterus, allowing the offspring to develop internally during the first 3 larval stages, in a method known as adenotrophic viviparity. During this time, the female feeds developing offspring with a milky substance secreted by a modified gland in the uterus known as the milk gland. In the 3rd larval stage, the tsetse larvae leaves the uterus and begins its independent life. It crawls into the ground and develops a hard outer shell called the puparial case, where it completes its morphological transformation into an adult fly. The larval life stage generally lasts 20 to 30 days, where the larvae must rely on stored food for sustenance. So that's the tsetse fly in a nutshell. But why are we covering Glossina fusepes in particular? Well, this species is a plague on sub-Saharan Africa, infecting both people and animals alike with pathogens. Pictured here is African Trypsomiasis, an insect-borne parasitic infection caused by the species Trypsomia brucei. The first image shows Trypsomia forms in a blood smear under the lens of a microscope, and the second is that of a 3D rendering of the parasite. Below these images is a graphic on the transmission of the parasite, or its life cycle, if you will. During a blood meal on a mammalian host, an infected tsetse fly injects metacyclic trypsomyogotes into skin tissues. The parasites enter the lymphatic system and pass into the bloodstream. Inside their host, they transform into bloodstream trypsomyosis and are carried to other sites throughout the body, reaching other bodily fluids like lymph spinal fluid and replicate through binary fission. The entire life cycle of African tryptosomes is represented by extracellular stages. The tsetse fly becomes infected with bloodstream trypsomyosis when taking a blood meal on an infected mammalian host. In the fly's midgut, the parasites transform into procyclic trypsomyosis, multiply by binary fission, leave the midgut, and transform into epizygotes. The epizygotes reach the fly's salivary gland and continue multiplication by binary fission. This whole cycle within the fly takes approximately 3 weeks, so pretty quick. African tryptomisis is commonly known as sleeping sickness and is the poster child of this fly's danger to humans. The trypanosome parasites infect the blood of their hosts, causing fever, weakness, and lethargy, leading to weight loss and anemia in the host, leaving some hosts with a fatal version of the disease. African tryptomisis occurs regularly throughout sub-Saharan regions of Africa, with the population at risk being about 70 million in 36 countries. In 2015, it caused around 3,500 deaths, which is considerably down from a total of 34,000 deaths in 1990. More than 80% of these cases are being reported out of the Democratic Republic of the Congo, which has been one of the locations of major outbreaks of the disease in recent history. Treatment for these diseases is completely dependent on the disease themselves. There is not one treatment for them all. African tryptomisis treatment is primarily done through oral medication. The specific medication depends on the disease stage, 1 or 2. The earlier the disease is identified, the better the prospect of a cure. If detected early, tryptomisis and other animals besides humans can be treated with tryptocidal drugs for therapeutic and prophylactic purposes. Here at the OHA, we are committed to global health and have researchers all around the world conducting studies and experiments to try and better understand and control this vector. Bedell and his team seek to use population genetics to aid in the implementation of vector control of tsetse flies in Uganda. Their research dives into a subspecies of Glossina fusepis. This subspecies is a riverine tsetse fly and is the main vector of human and animal tryptomisis in Uganda. Within the confines of the study, researchers conducted genetic analysis on mitochondrial and microsatellite data accumulated from approximately 1,000 individual tsetse flies captured in Uganda and the neighboring regions of Kenya and Sudan. Utilizing phylogenetic analysis, the data suggested that the largest scale genetic structure in the subspecies arose from a historical event that divided two divergent mitochondrial lineages. Further tests of isolation by distance suggested that there is ongoing gene flow between neighboring populations of the subspecies. Concluding their study was made clear that when planning for tsetse control in Uganda, a wider area encompassing neighboring regions must be utilized to achieve successful vector control. Gooding and Craftsworth's work takes a look at the contributions that testing and the monitoring of tsetse flies provided to the overall control of the species. They have identified 30-plus species and subspecies of tsetse flies that live within sub-Saharan Africa. This study reviews tsetse genetics from organismal and populational points of view and seeks to identify research needs. Genetic studies performed on tsetse flies within this study help to clarify systemic relationships among taxa and have provided important data on gene flow among the natural populations. In conjunction with the research, genetic maps were provided for the major species of tsetse flies within the region. The study concludes that further research should be conducted to study more definitively the mechanisms of sex determination, vector trypsomyosome adaptations, and the adaptation of tsetse flies to their various environments. Sleeping sickness is claiming more victims year after year, and genetic research could provide some relief in battling disease. Similar to the study conducted by Beadle, the researchers working on this apply the genetic diversity and population structure of the tsetse fly subspecies Glossina fusepis in northern Uganda towards the betterment of vector control. From here, it was identified that Uganda is the only country where the chronic and acute forms of human African tryptomisis both occur, and that Glossina fusepis is the main vector of the trypsoma parasites responsible for these diseases, as well as for the animal African tryptomisis. Using highly polymorphic microsatellite loci and a mitochondrial DNA marker, researchers working on the study are able to provide fine-scale spatial resolution of genetic structure of the subspecies from 42 sampling sites across the northern region of Uganda, where the two diseases are highly concentrated. Utilizing microsatellite analysis, researchers were able to conclude that the subspecies in northern Uganda are structured into 3 distinct genetic clusters. Taking these findings into account, the team identified Tsetse migration corridors and have recommended a rolling carpet approach for vector control within Uganda to minimize disease dispersal and prevent vector recolonization. Additionally, the findings provided by the research team highlight the need for a continuation of Tsetse monitoring efforts during and after control of the vector. OHA plans to do just that, to continue to monitor the tsetse fly and work to eliminate the threats that the vector poses to us. From here, we move forward with our management practice strategies to achieve that goal. For starters, we must continue researching tsetse flies and their associated pathogens in a lab setting as we seek long-term vaccines for the diseases they bear. From there, we have our boots on the ground approach, wherein we have resource groups supplying the people of sub-Saharan Africa with the means of protecting themselves from these flies. From screens on windows and clothing to cover the skin to the removal of tsetse habitat around populated areas, coupled with our in-person work, we lobby for the effective, accessible, and affordable disease diagnostic and treatment within the framework of a multi-disease control approach. This includes not only the governmental side of things, but a supply of medical professionals to aid those that need it. Well, that wraps up my time with you here today. So please, if you have any questions, now is the time. Thank you very much.